In this video, we're going to look at all the reactions that it takes to take pyruvate and turn it into acetyl coenzyme A. So we start with enzyme 1, pyruvate dehydrogenase. And the first step is to decarboxylate this pyruvate, so you remove a CO2, and you form a hydroxyethyl TPP intermediate. So here's TPP. We need TPP for this to work. And basically, you're just attaching pyruvate to TPP, and you remove a CO2. So you're removing this CO2 from it, and you get your intermediate. And that's step 1. Step 2 is that we're going to take this hydroxyethyl and transfer it to the next enzyme, dihydrolipoyl transacetylase, or E2. Now for E2, the important part of this enzyme is shown here. It has a Lippoamide group made of lipoic acid that is covalently bonded to lysine. And the reactive center for this group are these two cysteines, this cyclic disulfide. And these are reversibly reduced. And when you reduce it, you get dihydrolipoyl. What happens is that we get our TPP intermediates from our first reaction, and that's going to attack our cyclic disulfide here, breaking this sulfur-sulfur bond, and we get this intermediate structure right here. TPP is removed along with an acidic hydrogen, and we're left with our new intermediate, hydroxyethyl carb anion. And that is giving us this acetyl group on lipoamide disulfide. So no longer need enzyme 1. That's gone. Now we have our 2-carbon unit attached to E2. Reaction 3 is simply taking coenzyme A and adding it to our ethyl group, and you'll get acetyl coenzyme A. And we have made our product. We have made acetyl coenzyme A, so that's awesome. The only problem is E2 is broken now. So E2, remember, we used to have this disulfide bond. We don't anymore. So we need to reform our disulfide bond. And that's step 4. And enzyme 3, the whole purpose of enzyme 3 is just to reform the active site of enzyme 2. So that's what dihydrolipoyl dehydrogenase, or E3, is going to do. It's going to reoxidize dihydrolipamide, and that will complete E2's job. Now, the way that E3 does this is that it has a 2 cysteines that are in a disulfide bond, and it has an FAD plus, or sorry, just an FAD. So these 2 sulfurs are going to break apart, or more or less they're going to take the hydrogens from E2. The disulfide bond in E2 will be reconnected, and now E3 will be in its reduced form. And the last step is we need to reform E3 before we can redo another cycle. So we need to reoxidize it, and that's just simply NAD plus will come in and we'll make NADH. What actually happens is that this FAD will take 2 hydrogens from this sulfurs to make FADH2. You have a disulfide bond. Those hydrogens are then given to NAD to make NADH and acidic hydrogen, and E3 is ready to go, ready to do reaction 4 again. Now, the main player in all this is our lipoamide arm, our lipolysyl arm, which is made out of a lysine and a lipoamide group. And this is on E2. And this thing is actually flinging our substrate, or channeling our substrate, between enzyme 2 and enzyme 1 and 3. So in total, when it's connected to lysine, this arm is roughly 14 angstroms. This is further connected to a highly flexible greater than 140 angstroms proline and alanine rich segment, which is like a tether. So you can imagine that this E2 protein has this like long tether that is allowing it to connect to enzyme 1 and enzyme 3. And if you remember how this complex is set up, enzyme 2 is in the center and enzyme 1 and 3 are on the outside. So enzyme 2 is just kind of interacting with 1 and 3 using its long tether. And so what this means is that each E1 protein can acetylate a number of different E2 proteins, because these E2 proteins just have these arms that are reaching out to different E1s. And E3 also can reoxidize several E2s, because you have a lot of these proteins around, so they don't have to work with like specific partners. Any E3 can interact with any E2. Any E2 can interact with any E1. And that is it for this video. I will see you in the next video where we go over the citric acid cycle reactions.